Okay, good morning, everybody. It's my pleasure to tell you about some of the developments at Scancell today. I'll just draw your attention to our disclaimer notice. This presentation will be available for download from the Scancell website if you really do want to read that. Okay, so we are a differentiated immuno-oncology company. Now, it'll be no news to this audience that the immuno-oncology market is growing very rapidly, mainly on the back of the success of the checkpoint inhibitors. And whilst the overcoming immune suppression has really opened up a whole new range of opportunity for immune activation. And this is where Scancell comes in, and specifically around T-cell activation, with our two platform technologies, which we refer to as Immunobody and Moditope. So from those platform technologies, we have 4 lead products in development. Some of those products will be entering into clinical trials throughout 2019. And the company was founded by a renowned immunologist, Professor Lindy Durrant. We have our offices based in Oxford. We're about 23 employees today, and we are listed on the AIM exchange with the ticker SCLP. Okay, so this concept of immune activation by delivering cancer antigens is not new. There have been several cancer vaccine approaches, but have had modest results in the clinic. And we believe this is partly because of the selection of the cancer antigens or range of cancer antigens or cancer epitopes to those antigens, but also the method of delivery. And Scancell has overcome those limitations and happened to come up with methodologies to induce potent or high avidity CD8 and CD4 T-cell responses. And we do that in 2 separate ways. So with our Immunobody platform, it's a unique mode of delivery that allows the processing of these cancer antigens into a dual mode of action that induces very high avidity CD8 T-cell responses. Now, in the case of our Moditope platform, this is a whole new class of cancer vaccine approach. And this is by the identification of highly immunogenic antigens that are basically produced through stress-induced post-translational modifications. That's quite a mouthful, so we'll call them symptoms. And Scancell is the first company to come across with this concept and to take these forward into the clinic. So this is our development pipeline. So our lead program is an Immunobody. This is a product we refer to as SCIB1. It's directed towards malignant melanoma, so it's been through a phase 1/2 clinical study in about 35 patients, so a cohort with malignant disease and a resected patient population. And we've been able to show efficacy in both of those patient populations, as well as a very favorable safety profile. And we're now advancing that product into a phase 2 clinical study in 2019. Our SCIB2 product is in partnership with CR UK, Cancer Research UK. So they are taking this product into evaluation in the clinic, again in combination with a checkpoint inhibitor, to evaluate its safety and efficacy in non-small cell lung cancer. And Scancell has an option to bring that asset back into the company at the end of that study. Our lead Moditope program is Modi-1. And as I said, this is our first symptom vaccine. And that will be going into the clinic next year in a range of cancer indications. Modi-2 following behind, going through preclinical evaluation. And then on top of that, we have a collaboration with BioNTech. This is Europe's largest private biotechnology company based in Germany. And we have a collaboration with them to identify the T-cell receptors. So that's receptors on T-cells that can identify our Moditope antigens. So this will be going into the adoptive T-cell therapy approach. And then earlier this year, we brought in some therapeutic antibodies targeting sugar signatures on cancer, and we're currently going through lead optimization of those antibodies to take them further forward. Okay, so we referred to the Moditope platform. So what do I mean by stress-induced post-translational modifications? So stress cells undergo this process of autophagy or autophagy. And this is the cellular processing of proteins. And during that processing, we get these transformations. So as these proteins are broken down, there are a couple of, or many transformations that occur. And 2 of particular interest to us is the conversion of arginine to citrulline, which is mediated by PAD enzymes, and also the conversion of lysine to homocitrulline, which is a chemical reaction, carbamylation. And what happens in a cell when it goes through this process is that the homocitrulline and the citrulline fragments can be presented on MHC class II and induce very potent CD4 T cell responses, cytotoxic CD4 T cell responses. And the step that Scancell took was to essentially identify, characterize, and immunize with these unique antigens to induce very potent CD4 T cell responses for the treatment of cancer. And we have very strong patent position around the utility of citrulated and homocitrulated peptides in the treatment of cancer. So, case in point, so our first program, as I mentioned, is MODI-1. That will be based on the citrulated antigens, and MODI-2 will be focused on homocitrination. So MODI-1, the first program to go towards the clinic, consists of 3 peptides, citrulated peptides, 2 of those coming from Vimentin and 1 from enolase, and both Vimentin and enolase are highly upregulated in cancer. So if you look at some of these very hard-to-treat cancers, for example, triple-negative breast cancer, ovarian, and sarcoma, you'll see very high expression levels of these proteins in these cancers, as well as upregulation of the PAD enzymes, which convert the arginine to the citrulline. And if you look at this mouse model at the bottom of the slide, you'll see that if you immunize the mice with either the Vimentin or the enolase peptides, we get very good survival rates of around 80 to 90%. However, if we combine the two, we get 100% survival. Now, we've repeated this in many other models. We've also re-challenged the mice with the tumor, and we've got spectacular results. And that's obviously led towards taking this product into the clinic, which I mentioned will be initiated in 2019. So the focus here is to do a dose escalation, assess safety and tolerability, and then to advance the product into 3 cohorts of patients, currently looking at triple-negative ovarian and sarcoma individual cohorts as a monotherapy to assess efficacy. And the finished product, actually, with these peptides, we conjugate with an adjuvant. And the adjuvant we're using is from ICER Pharmaceuticals. We'll hear more from ICER in the next presentation. And they have a TLR1/2 adjuvant, Amplivant, and each of these peptides is conjugated to the adjuvant and combined as the final product, which we refer to as MODI-1. And that allows us basically to boost their immune response from 10 to 100-fold. And so instead of using milligram quantities of product, we're now immunizing with microgram quantities in this study. We're also, as I mentioned, working with BioNTech to identify the T-cell receptors against these moditope epitopes. And the idea here is to, obviously, once we can identify these, is to convert those to a vector, which can then be transfect the T cells isolated from patients and expand that as an adoptive T cell therapy approach. And the advantage is that because we see these citrulated and homocitrulated epitopes in a broad range of cancers, this gives us a massive opportunity in terms of how we might use this adoptive T cell therapy approach across cancer, but also that these antigen-specific T cells are recognized as non-polymorphic HLA-DP4. So this is applicable to the vast majority of patients. So just to summarize on a moditope, we've obviously, as I mentioned, got this relationship with BioNTech to identify T cell receptors. We've in-licensed the ICER pharmaceutical Amplivant technology. We're now in GMP manufacture, so we have our GMP manufacturing process and just about to enter a pilot manufacture for tox studies. We anticipate starting our MODI-1 clinical study in calendar year 19. Currently undergoing evaluation of the homocitrulated peptides for incorporation into MODI-2. We have a very strong patent position. And this technology was also the core component of a proposal to CR UK Grand Challenge earlier this year. We were shortlisted for that award, and CR UK will be announcing the winner of that award early in the new year. So now just switch track now to the Immunobody platform. So this is another proprietary platform to Scancell. And in this case, what we do is to integrate T-cell epitopes into an antibody framework. Then the product is delivered as a DNA plasmid using electroporation. So at the site of injection, you transfect both the muscle cells and the antigen-presenting cells. And that allows for the transcription to this antibody-like molecule that has two modes of action. So you get direct presentation to the antigen-presenting cells, but also cross-presentation via the Fc receptor on the surface of those cells. And that induces very potent or high avidity CD8 T cell responses. In the case of SCIB1 for melanoma, we're targeting TRP2 and GP100, so there's epitopes CD4 and CD8 epitopes of each of those antigens integrated into that antibody molecule. And then in the case of SCIB2, which, as I mentioned, partner with CR UK, we're targeting NY-ESO1. So this is some of the clinical data from our previous clinical study with SCIB1 in melanoma. As I mentioned, we treated 2 cohorts of patients, 35 patients in total, 15 of those with measurable disease and 20 patients with resected, so we're in the adjuvant setting as a monotherapy. And we saw responses in those patients with metastatic lesions in the lung, and we see out there on the left-hand side of the slide. And in the resected patient population, you can see from this swimmer plot, we had some spectacular results in the prevention of disease over a period of 5 years. So this swimmer plot shows the lead-up or each patient in the 5 years in the lead-up to the study, which is the black vertical line. And you can see that post-immunization in those top 10 patients on the horizontal lines, we were able to prevent the recurrence of disease for a period of 5 years following the immunization. So this is obviously pointing to a long-term memory response that's induced by the SCIB1 product. So we are also evaluated how we might be able to utilize SCIB2 in conjunction with checkpoint inhibitors. So we're about immune activation, checkpoint inhibitors overcoming immune suppression. So there's a nice synergistic mode of action between the two, which we, again, heard from the previous speaker. And when we actually look at this mouse model, you can see that both the checkpoint and SCIB1 have similar rates of survival in this model at around 60%. However, if we combine the two, we get a real boost and advantage of this synergistic mode of action to boost that response rate up to about 85%. So our objective now is to advance SCIB1 into a combination study. We'll be evaluating in 25 patients. This study will be led by Keith Flaherty at Mass General in the United States. It will be under an IND. We anticipate starting the study in 2019. And to evaluate the clinical relevance of SCIB1 in conjunction with standard of care, which will be Keytruda in this case, so combination. And so the idea here is that the response rate from Keytruda is about 30%, and we anticipate to boost that overall response rate to about 55% plus. So our identified opportunities obviously are in adjuvant setting and also in combination with our SCIBs. And in MODI, we're converting cold tumors to hot tumors and looking at these immunosuppressed tumors, if you like, to induce immune responses. Our highlights for our last year have all been milestones directed towards advancing these products toward the clinic. The company raised some capital early this year on the AIM market, about £8.7 million. And those funds will be directed towards the initiation of these studies as we step through 2019. So that's the SCIB1 study and then the initiation of the MODI-1 study as well. And this is all about identifying the clinical relevance and clinical value of our products, the eligible patient populations, and then from that data, then we can establish the commercial utility of our program. So these are, I believe, very huge value inflections as we start to understand the commercial opportunity that our products have. So in summary, 2 platforms, 4 lead products, 5 core activities. First and foremost, generation of clinical data, expanding our pipeline, leveraging our IP, looking for technology partnerships to de-risk our programs as we go forward, clinical partnerships to understand the clinical utility of our programs in real clinical practice, and then dialogue with those industry partners who have immuno-oncology franchises and are looking to protect that franchise or expand it with the optimum combinations. Thank you.